Hey guys, today we're going over meiosis, which is the type of cell division that makes gametes like sperm and eggs. Unlike mitosis, which makes two identical diploid cells, meiosis produces four genetically different haploid cells in most cases. Before we start, two key terms matter. Sister chromatids are identical copies of a chromosome made during DNA replication, held together here at the centromere. Homologous chromosomes are a matching pair, one from each parent, with the same genes in the same locations, but not necessarily the same versions of those genes. So these two would be sister chromatids, whereas these two would be homologous chromosomes. So meiosis I. In Prophase I, homologous chromosomes pair up tightly in a process called synapsis, forming pairs called tetrads. While they're paired, crossing over can occur, as we can see right here. Non-sister chromatids exchange matching DNA segments, and this is a major source of genetic variation because it creates new combinations of alleles on each chromosome. Next, in metaphase I, homologous chromosome pairs line up in the middle of the cell. The important detail is that each pair lines up with a random orientation towards either pole. That randomness is independent assortment, and it shuffles which maternal and paternal chromosomes end up together in the same gamete. In anaphase I, the homologous chromosomes separate and get pulled to opposite poles. Here's the key difference from mitosis. Sister chromatids stay together. The centromeres don't yet split. In telophase I and cytokinesis, the cell divides into two haploid cells. Each cell has one chromosome from each homologous pair, but each chromosome is still duplicated, meaning it still has sister chromatids. Meiosis II. An important detail here is that there is no DNA replication between meiosis I and meiosis II. The chromosomes are already duplicated from earlier. In prophase II, chromosomes condense again, and a new spindle forms in each haploid cell. In metaphase II, chromosomes line up one by one in the middle. This time it's not homologous pairs because those were separated in meiosis I. In anaphase II, sister chromatids finally separate and move to opposite poles. This is the step that looks most like mitosis. In telophase II and cytokinesis, each of the two cells divide again, producing four haploid cells. The final outcome is four haploid gametes. They are genetically unique because of crossing over in prophase I and independent assortment in metaphase I. One more major source of variation happens after meiosis, random fertilization, where any one sperm can fertilize any one egg, multiplying the number of possible combinations.